everyone, and welcome to this Bozeman video that's not a Bozeman video because I'm Elise, not Bozeman. Alright, so my topic is gene therapy, and first of all, what is gene therapy anyway? I don't know. Actually, I do, because I had to research this for this class. Gene therapy is actually an accepted therapy for the treatment of a disorder by manipulating an organism's genes. Okay, so what does that all mean? Manipulating an organism's genes? Well, basically, what biologists are doing is they are taking either viruses or liposomes and using them as transportation devices to insert healthy genes into a body to correct their disorder. Alright, so there are two types of ways to do this. One is in vivo gene therapy, and the second is ex vivo gene therapy. Alright, so in vivo is when the gene is delivered directly into the body, and ex vivo is when the gene is inserted into cells that were removed from the body, that are then returned to the body after the gene has been inserted. Alright, okay, so for an ex vivo example, let's say you have a familial hypercholesterolemia.
Familial hypercholesterolemia.
Familial cholesterolemia. Okay, yes. Basically, this disease is liver cells lack this receptor protein for removing cholesterol from the blood. So patients can get like heart attacks and stuff, and that's not good. So basically, for ex vivo gene therapy to correct this problem, a piece of the liver is excised. It is infected with a genetically modified virus so that the virus is safe, so that the virus contains the gene that has the receptor protein for cholesterol, removing cholesterol, and then the liver piece is then reinserted into the liver, into the body, and the patients are actually fine after that, usually. Most of the patients don't experience any more problems after that, which is really cool. It's actually successful. Another example is other sites of ex vivo gene therapy are the skin, endothelium, which is like the blood vessel lining, and then bone marrow as well. Alright, for in vivo therapy, an example is people with cystic fibrosis, which we all know what cystic fibrosis is because Ross loves to talk about it. They need a gene for the transmembrane carrier of the chloride ion, which is why they get like all phlegmy and they like cough a lot. Okay, so basically that gene that has the transmembrane carrier is then inserted into the patient either with like spray in the nose or into the lower respiratory tract. It actually has gotten limited success, which is why, I'm more excited about ex vivo gene therapy because the example they gave in the book is very successful, rather than in vivo. It seems more kind of like trial and error. Alright, another thing that could be cool in the future is in cancer patients. They're working on using the p53 gene to encourage apoptosis in cancer cells, and either make normal cells more resilient to chemotherapy and cancer cells more vulnerable to chemotherapy, which I think is really cool. But then again, in vivo therapy has had limited success compared to ex vivo therapy. But yeah, gene therapy is really cool. It's kind of controversial, but in the end, it's just a way to help people, right? So yeah, the next thing we're going to talk about is Genomics, which Kayla, my partner on this, is going to talk to you about. I hope this wasn't too rambly for you, but yeah, thanks.
All right, so the wrap-up of Genomics is going to be kind of long, so I'm sorry, but yeah. All right, ready? Keep doing this, and I'm really sorry. My voice is dying. Okay. Researchers know the sequence of the base pairs along the length of the human chromosomes. So far, researchers have identified around 25,000 human genes that code for proteins. The rest of our DNA consists of regions that do not code for a protein. Currently, researchers are placing an emphasis on functional and comparative genomics. But, you know, this is also a textbook and I have no clue how old this is, so. Genes comprise of only 1.7 of the human genome, and the rest of this DNA is surprisingly more active than once thought. About half of this DNA consists of repetitive DNA elements, which may be in tandem or intersped throughout several chromosomes. Some of this DNA is made up of mobile DNA sequences called transposons, which are a driving evolutionary force within the genome. The role of the remaining portion of the genome is actively being investigated, but it's hard to believe that these DNA sequences may play an important role in regulation of gene expression, thus challenging the classical rule in regulation of gene expression. See, I told you guys, I'm tired. Alright, thus challenging the classical definition of the gene. Thank you, Kayla. Man, guys, if you want to turn me into a meme, go ahead, because this is, like, ridiculous, and I'm sorry. There's a lot of stuff wrong. Just, yeah. Ross, you know about this. Anyway. To that end, researchers are utilizing tools such as DNA microarrays, and then microarrays can also be used to create an individual's genetic profile, which is becoming helpful in predicting illnesses and how a person will react to particular medications. Comparative genomics has revealed that little difference exists between the DNA sequence of our bases and those of many other organisms. Genome comparisons have revolutionized our understanding of involuntary relationships by revealing previously unknown similarities between organisms. Proteomics, or however you say it, someone is going to be like super pissed with me because I can't say it. Proteomics is the study of which genes are active in producing proteins, in which cells under which circumstances. God, Kayla, really? All right. Bioinformatics is the use of computer technology to assist proteomics and functional and comparative genomics. Guys, we are done. That was forever and really, really long, and thank you if you did stay all the way to the end of this video. You are brave. You are very, very brave and have time on your hands. But thank you guys for watching this. I really do hope that helped. It might have not, and I'm sorry about that. I'm sorry about my voice, and I'm sorry about having to stop because of the pain I'm in. It's starting again. Alright, I'm gonna leave off on that note, you know, that wonderful note of pain. Yeah. Thanks for everything, guys. Bye. Hopefully I don't have to do this ever again. Yay.